face-to-face exchange of experience of conservation between Mexico and Australia. And hopefully it comes out with great collaboration, information, research, and just amazing results. We'd love to learn more about the research that's going on in both countries, how they relate, and also, of course, how to learn from each other, obviously. I think that will be amazing. And then the most important one is there's always a challenge when it comes to research, when it comes from different cultural backgrounds and or different landscapes and such. And I think this is a perfect opportunity to show and talk about those challenges, how to overcome it, and actually how to make it an opportunity. Now, before I would like to directly continue to our first speaker who'll talk more about this side from us in UQ and the work that we do over here in the Center of Conservation. Dr. Daniel Dunn will be taking this. So Dr. Daniel Dunn is an associate professor with us in the School of Environment, and he is also the director of our Conservation Center at the University of Queensland. His research focuses on how migratory species and connection with the ocean, and also on spatial management measures to conserve and how we can work together in regional and global scales. Daniel has also worked with 7 UN conventions and organizations to try to provide information necessary for a healthy ocean. Daniel also is an amazing person when it comes to engaging with researchers alike, many level ECR, PhD students, so much support, lovely. And we're gonna hear a lot more about him and his work. So I will just send it over to Daniel. Please take the floor. Thank you.
Hi, Carlina. Thank you so much. Hopefully everybody can hear me okay. Okay. Hola a todos. Me da mucho gusto de introducir esta conferencia científica. I started some of my research, my very early days in Baja California del Sur. And it gives me great pleasure to have an opportunity to sort of continue to facilitate exchange. I am from the United States, but I've now moved to Australia and been here for a few years. So I'm glad that even though I've moved across the Pacific, the exchange between scientists will continue, and that we are building these bonds. So with that, I would like to share my screen, and I will give you a brief introduction of the Center for Biodiversity and Conservation Science. Hopefully you can see that screen. Yes?
Yes, we can see it.
Thank you very much. Okay, I'd also like to start by acknowledging the traditional owners. We've already done the formal acknowledgement, but just to acknowledge country across the globe and the traditional owners in Mexico as well. It's really critical and a growing theme here in Australia, regardless of certain elections or votes that we've just had. This remains an absolutely critical issue for the center and for the university. And I'm very excited that this is sort of part and parcel of the discussions today and ongoing work between all of our researchers. So I just want to give you a brief overview. The center has about 35 affiliated researchers. We have a number of sort of laureate and future and DECRA fellows, which are sort of fellowships across the spectrum here in Australia, from the sort of preeminent scientists in Australia, the laureate fellows, to all the way to the early career researchers. And we are very successful in getting these kinds of fellowships. We also have the former, what we call pseudo-retired chief scientist of Queensland here, which is Hugh Possingham. He has been a key part of the center for many years, and it's great to have him on board and be able to feed our information, feed our research directly into the work that the policy and management issues in Queensland. through him and through others doing similar types of work. And I look forward to hearing how our Mexican colleagues are able to sort of feed their information into the policy arenas there in Mexico as well as more regionally and globally. Probably more important for this workshop and facilitating things is that we have over 100 PhD students and 30 postdoctoral researchers, Carlina being one of them, and all of this has been set up essentially by PhD students and postdoctoral researchers who are really the main engine of the center and drive all of our sort of workshops and the key efforts that we have around developing new initiatives. So thank you very much, Carlina and Evelyn, for all of your work in putting this together. A brief summary of CBCS. We're about 13 years old now. We have many global partnerships, so very excited to see what we can develop with our peers in Mexico. Last year, we had about 220 peer-reviewed publications, so the main currency of academics. So a lot of publications, which have resulted in a number of articles in sort of Nature Index journals, and the #1 position for in Australia for the 2022 Nature Index in this arena. So just obviously there's a lot of focus on publication and a lot of focus on bringing new research grants in, but it's not just that. Really, the main point of CBCS and the thing that binds all of us together in the center is the applied nature of the work we do. So I just want to spend a couple of minutes going over more that side of it rather than the publication side. So I'll give you a couple of examples of how we've sort of engaged with policy arenas. The first is that on a national level, we have something called the State of the Environment Report. And we went through the last State of the Environment Report because we were curious how much of our work was feeding into it. How many times our affiliated researchers or PhD students or postdocs were authors, and the level of citations in this sort of critical national report. And it turned out that actually CBCS was more highly cited than any other institute in Australia in feeding into this. We co-authored for the sections, and we developed some indices that were actually used in this report as well. So just really very strong integration between our work and the sort of national policy relevance and how it's being taken up within national policy. But it goes beyond that as well. We did the same sort of thing looking at the IPBES Biodiversity and Ecosystem Services assessment, the global assessment, and found that there were over 100 authored or co-authored papers by CBCS researchers across all of the chapters in this major report. And again, that's just sort of very obvious to us that we're succeeding in our main goal of trying to feed this information up into policy arenas. And again, look forward to seeing how this sort of burgeoning relationship with Mexican colleagues can result in sort of a pan-Pacific approach to this as well, and feeding into other policy arenas. And then finally, I want to say that it's not just at those levels, it's across a lot of different policy arenas. So at the bottom here, I'm talking about other policy arenas that we have been participating in. But the thing at the top about the jointly funded positions is really the most important thing here, where we work very hard to find these sort of co-positions, jointly funded positions with NGOs and with sort of semi-governmental organizations, which is what allows us to feed this information into policy arenas so much. So we've had jointly funded positions in the last few years with the Wildlife Conservation Society, with the World Wildlife Fund, with Nature Conservancy, and with CSIRO, which is sort of a major sort of semi-governmental scientific organization here in Australia. And that, again, is sort of what allows us to feed into all of these other policy arenas and management arenas. So a few of the topics that our researchers cover, there's been a lot of push in the last few years around environmental markets and blue carbon and ecosystem restoration. We have a very long history of thinking about conservation planning. The tool MarkSand was developed by Hugh Possingham and Ian Ball, and really sort of came into its own through researchers at UQ, driving that and feeding that into the Nature Conservancy and their work, but also thinking more broadly about conservation planning and decision science. So of all these things, there's actually one that I really want to emphasize here, which is globalizing conservation research. This is obviously the topic of one of the topics of this conference, and it's a really critical area of research for us over the last few years. And it's not just the work of particular researchers like Tatsuya Amano and others who have been sort of leading in thinking about how English is a barrier to research and the impacts that has on researchers where English is not your primary language. But it actually goes down within CVCS to what we're trying to do for our students. We recognize that we actually have a lot of students from Central and South America coming over, and that the process of participating in research in Australia can be very difficult for a number of reasons. One, the language barriers, but more beyond that, also just cultural barriers and trying to understand it. And that those mix of cultures are absolutely wonderful in terms of the breadth of thinking that we bring to the table in one place, but there are also barriers and obstacles that come with that. And we have tried very hard through our small grant scheme, which resulted in this workshop, but also resulted in other efforts to have surveys for our HDR, our PhD students and postdocs to understand what those barriers look like, and then begin to work with the university, and not just our university, but other universities in Southeast Queensland to try to address those barriers. So I was very excited to hear about this conference proposal and the effort to translate, because puedo hablar español un poco, pero mi español es como un chico de 15 años. Si necesito pedir una cerveza, puedo hacerlo, pero no puedo hablar científico. Y por eso es muy importante que tenemos translación, and that we're able to find ways to engage across languages for everybody, even those of us who can't speak Spanish like a 15-year-old. So much appreciated for the opportunity to make this introduction. Very excited about this workshop, and would love to see further progress on this afterwards and think about what the next steps are and how we can keep this engagement going. So thank you very much for coming, and for so many of you being there. It's amazing, and we look forward to the conference and to the next steps. Thank you, Carolina.
Thank you, Daniel. Really interesting talk there to listen about what's been done from the UQ, from the Australia side. After Daniel, we are now actually would like to welcome Dr. Abram from UNAM, who will be talking about research from the Mexico side. And then after that, there'll be one more talk with Dr. Matthew Luskin, and hold your questions till the end of the three speakers, then we'll open Q&A after that. So I will give it to Dr. Abram. Please take the floor. Thank you.
Hi, can you hear me? All right, everyone?
Yes.
Yes, perfect.
Oh, great. Now I will ask to the team here to put my presentation. Share the screen. Okay, I will start. There in Australia, but also here in Mexico at UNAM. And I would like to say that our university is a huge university. It's not like a small center. We have like this small faculty, and I will address that a little bit. But also I would like to talk about the UNAM in general. We're part of the network of macro universities. We have around 350,000 students, from undergrads to PhD and master students, and also for high school students. So we have like this huge basis of students at our university. Please, the next slide. I hope all you guys are looking at the presentation, because they already have it here. No, that's not. I don't think so. Yeah, that one. So please, next slide. So as I told you before, we're like huge university. We have 373,000 students from the high school system to the postgraduate student studies. But also we're the 93rd university according to the World University Rankings. So we're like a huge university, but also a good university. And as you can see, we have like also a lot of places to study, and this is over 2 million of square meters for research, culture, and teaching. Please, the next slide. So at UNAM, we have like these two subsystems. One of them is the scientific coordination, which comprises 24 institutes and 6 centers devoted to scientific research. We also have another side which is for humanities, but I will just cover scientific research now. And this hosts 6 programs and manages 3 natural protected areas, and co-manages another one in the state of Tlaxcala. So in order to do field work, we also have like a lot of facilities. Please, next slide. And as a big university, we have a general direction, which is in charge of cooperation and internationalization, with coordination for international affairs. And as you can see, we have different places to facilitate the interaction with different countries, mainly on the US and Canada, but we also have places in China, Germany, UK, France, and Spain. And recently we opened just one in Cuba. Also, we have a lot of students exchange, and usually per year, pre-pandemics, we have like 400 students going abroad, mainly to South America and North America, but we also have a lot of interaction with European universities. And as you can see, we have like this huge gap in Australia. So I think we could start filling that gap. So pre-pandemic, we have all those students going abroad, usually around 900 for the whole university. Considering our faculty, usually it's around or 80 from different careers. But now with the pandemics, we have like a huge reduction, but now it's going up again. Please, next slide. So regarding our faculty, I will talk on behalf of our dean, headmistress, which is just beside me. And I will talk you a little bit about our faculty. We like to do like the best of our work to achieve in the formation of excellent students, to have a comprehensive, high-quality education in the fields of health, environment, and life sciences in general. This is the next one. And we are a multidisciplinary faculty, which means that at our faculty, we have students from the bachelors of medicine, dentistry, nursery, optometry, psychology, ecology, and biology. Our faculty also has like big facilities. We have over 22,000 square meters in buildings at the main campus, and also 16 facilities around the UNAM, mainly with clinics to provide services for dentistry. We have a lot of infrastructure, which comprises the, as I've told you before, the clinics for dentistry, optometry, but also we have some places dedicated mainly to research. And we also have library, bookstore, auditorium, gymnasium, etc. We are a pretty old faculty. We're almost 50 years old, and it started like a small program for decentralize the education that has been mainly provided at the southernmost part of Mexico City. And as a faculty, we have different or participate in different postgraduate programs. So this is our bachelor's degrees, and we have around 18,000 students at our faculty on the different levels. So what I'm just trying to say, please, the next one, is that we have a lot of students going abroad, and many of our students go to Central and South America and also to Spain in Europe. And this is usually because of the barriers that languages represent. However, we're sure and certain that we have to start working with different partners as the University of Queensland in order to start changing this, because as long as we start expanding our view on how the world works and how our students can actually learn from different people with different cultural backgrounds, we will improve the education that we're providing. So we have 4 research units, and I will emphasize the work that they do in 2 of them, which is the unit in research in morphology and function, and also the research in biotechnology and prototypes, where we have several researchers working with different approaches to conservation, ecology, and management. And all this is organized in a bigger program that we'll start launching, which is called FEST-INT, which means that it's our faculty and our efforts to actually internationalize the knowledge and also provide the students with different opportunities to learn from different cultures, and in that way improve their formation. So I just like to finish providing like small information, and I like to thank, yes, our team here and also the team that is working there really early in the morning, and also the students for staying here this late, because usually they go earlier. And I like to thank also Maria del Rosario Mata Peña Jaramillo, the minister from Mexico in Australia, and the Center for Biodiversity and Conservation for their small grant scheme that provide us with the funding for doing this workshop. And we will be very, very thrilled to look for different ways to interact with you and also provide like the basis to make work, because in 2004, our universities started working on an agreement, but sadly, it never actually launched. So I think that almost 20 years later, we make it happen. So thank you so much for listening, and I'm really eager to see the next talks. Thank you.
Thank you. That was very, very interesting. I'm just going to go directly to the next speaker, Dr. Matthew Luskin. Feel free to take the floor. Thank you.
Hello, everybody. I am tuning in from a different CBCS event. This is the Early Career Researcher Workshop. We are on a beautiful island in the Great Barrier Reef called Heron Island. I'm actually here with Carlina, but in a different room. I'm going to share screen and get started. Okay, you should see how unorganized I am. And now I go to full screen. Are we looking good there?
Looks perfect.
Okay, cool. All right, so I am a wildlife ecologist. I focus on trophic cascades in Southeast Asia. That means what happens when large predators are removed from ecosystems. But today we have a special presentation because I will talk about tigers. And then a recent graduate student, Alejandro Garza Garcia, who just finished his master's. Oh, it says Alejandra. Dude, sorry, man. It's Alejandro. He just finished his master's here at UQ in our conservation biology program and did a whole study of jaguars. So I'm going to do about 9 minutes, and then I'm going to hand it over to him to do 9 minutes on jaguars. Okay. So just to orientate us, this is Southeast Asia. This is showing plant diversity. It's a very, very big biodiversity hotspot, but there's relatively few research papers in ecology in this area. It's also, this is a number of threatened mammals, very, very high levels of threat due to hunting and habitat fragmentation predominantly. This is the forest cover. The pink shows deforested area. You can see the remaining habitats are very fragmented. And also the reason for that is largely in the last 25 years, the growth of one crop, oil palm plantations. These are used for vegetable oil. That's the photo you see on the right is a bunch of oil palm fruits growing out of the base of the palm tree there. It's really productive. The only downside to oil palm is it grows where tropical rainforest grows. So I'm going to talk about a study we did in Sumatra to look at the tigers. And so this is the habitat availability in Sumatra in 1985 versus 2010. You can see how much of it has been lost relatively recently. In fact, 50% of the forest was cut down in just 25 years. So we decided to focus on tigers because that's who gave us money. I wrote the exact same grant to study other animals and it was not funded, but people love tigers. You can also make a case that they're potentially really important. So this is their place in the food web in Southeast Asia. They may be a keystone species. We are testing that right now in a different project in the lab. So we had a few questions here. One, what are the factors influencing tiger densities in Sumatra today? A lot of people have said they see a lot of tigers in plantations. Tigers are eating livestock out of plantations. So we asked, are plantations suitable habitat? And the way we tested that is, are the densities in plantations similar to the densities in forests? Which remaining forest patches have viable tiger populations? And viable here means 25 breeding females to prevent genetic inbreeding. What's the total population size? Everyone wants to know that number. And is the trend increasing or decreasing? So this is where we did our study. We sampled these 3 very well-known national parks. These are also make up the UNESCO World Heritage Site Rainforest of Sumatra. We put out between 60 and 90 cameras at each one. These areas cover 500, 800 square kilometers. And then we got lots of pictures because what we were doing is we were setting camera traps. There's a picture of us setting cameras dressed as Rambo. We get pictures of all sorts of animals. So our study species, tigers, of course, but then all sorts of cool, cute critters. That was a tapir. These are elephants right before they smashed the camera. Can you guys see what's here? Clouded leopard, beautiful animal. Lots of pigs until recently, since now there's African swine fever, all these pigs are dead. So many pigs, they get piled up. Sun bear, pondering life. A baby sun bear. Some selfies. Porcupines, they always travel in twos with their partner. Some mating displays from the Argus pheasant. Pretty cool. Even some evidence of tiger attacks. So you see that big claw mark on the tapir's shoulder there. All right, so we were looking to estimate densities across these landscapes, comparing parks and their relative abundance within the parks. So we use the stripes to identify each tiger. Then we use mark recapture and a version of mark recapture called spatial mark recapture to estimate their densities while accounting for the probability that we detect them. I'm not going to go into the stats, but how you study tigers ends up being as important as the actual factors affecting tigers. And that's something that Alejandro is going to talk about for JAGs in a minute. So what do we find? Well, these are our 3 parks. And the first thing to notice is we have huge variation among the parks. Okay, so twofold differences in densities. The second thing is to look at the second category, that's the second row there, and that shows that plantations on average have 71% lower density than forest. So that means tigers are using plantations. They're hunting, they're moving through them, but it's not high quality habitat for them. But it does mean they're probably not dramatic blocks to migration and connectivity. We also found that they use huge home ranges, much bigger than in India and other places. And that's important because it means we need larger reserves to conserve Sumatran tigers than we need for other large carnivores potentially, or even tigers in other areas. So then we wanted to know, our next question was, all right, this is good. We have some densities. We see some variation, but how do we know if this is a normal populations densities for tigers or if it's changed through time? So for that, we needed to compile all the previous density estimates, and then we need to standardize them because people use a lot of different methods. And then we use some regressions to look at which factors assess impact tiger densities. And particularly, we're looking at whether or not their densities are changing through time. So these are the results of that analysis. We got, I think, 18 different density estimates. And what you see at the top there is that time since 1996, when the first density estimate came out, the density, the effect on density, which is shown on the x-axis, is positive. What that means is through time, we've seen higher and higher tiger densities. And that is not surprising because there's been a massive investment in conservation efforts and anti-poaching patrols in Sumatra. And that shows that it's likely been effective. But we see that areas where there's a high deforestation rate have lower tiger densities. That's also likely because that's where people are actively hunting. Because where there's deforestation, there's new roads, people can come in and hunt. We also see that they are less likely to be abundant in secondary forests. These are selectively logged forests, and in relatively unproductive habitats, such as peat and montane forest. So then if we look at all the different habitats there, we can use these densities for the different habitats to estimate the population size in each park. And when we do that, and we do that for the year 2000 and the year 2012, we have these 15 key parks, and we find that there's been an 18% decline in the total number of tigers over the last 12 years, even though the densities have increased. And the reason for that is that the deforestation rate has outpaced the rise in tiger densities. Okay, so to answer our questions, are tiger populations increasing or decreasing? They are decreasing, even though densities are increasing. We're packing more tigers into smaller parks. Are plantations suitable habitat? Sort of. Lower densities in the plantations, but they are using plantations. What are the key drivers of tiger densities today? Deforestation, likely because of ongoing poaching. And what is the wild population size in Sumatra? 618. Compare that to well over 6,000 Sumatran tigers in Texas alone. So very few wild Sumatran tigers, but plenty in captivity. We also identified the viable tiger populations remaining in Sumatra. And our next steps, this is what the lab is focusing on now, is looking at what are the different outcomes for losing predators. So sometimes you have hunters, they take out all the predators and large animals, and often that will lead to increases in rodents and other things like that. Sometimes you lose just the predators, and then you have increased abundances of the prey species like pigs and deer. That can have negative influences because they overconsume the vegetation. That's a top-down predator cascade, or known as a trophic cascade. And we're also looking at the impacts of bottom-up fruit subsidy cascades. And this is when you have more fruiting, or a crop like oil palm producing extra fruit that some of the animals can eat, and they become more abundant, and that could support more predators. So these are the ongoing questions in our lab. Okay, I'm going to say thank you, and I'm going to hand it over to Alejandro. So I go stop share, and I'm going to mute.
Hola a todos, buenos días y buenas tardes. Gracias, Matthew, por la presentación. y yo les voy a hablar de mi tema de investigación y por si no delataba el apellido, soy de Monterrey. Okay, so I'll be talking about estimating jaguar densities while controlling for methodological bias. Jaguars are the third largest cat species in the world and the only living member of the Panthera genus in the Americas. While unlike their Asian and African cousins, jaguars are globally listed by the IUCN as near threatened. And while we have good data on well-studied subpopulations, globally, we don't have comprehensive information on their densities or the population trends throughout their range. And this is of concern. Like tigers, jaguars are apex predators. And given their affinities with water, jaguars affect systems, aquatic systems, as well as terrestrial ones, making them an interesting keystone species and an interesting species for conservation. After tigers, jaguars are the most common study species in camera trap research. Being a key figure in this field, jaguars are also quite commonly used in capture-recapture studies. In this context, capture-recapture studies work by establishing a survey to detect the species with camera traps. By processing the image, we can use the cat's unique code markings to identify unique individuals. With these individual IDs, then we can create capture histories for individuals across the survey. With these capture histories, we then can create a population size estimate using a capture probability times the initial captures. And for density, we use that said population size estimate divided by the area comprised of the convex polygon where the camera trap array is, shown here in the light gray, and adding a buffer. Now, in classic capture-recapture studies, that buffer is estimated using the mean maximum distance move within the camera array. And this can create some issues. Firstly, it's non-spatial. It's quite easy to underestimate home range size. And with a smaller buffer, that means that we have a smaller overall area where we detect the species and produce higher density estimates. With spatial capture-recapture, as the name implies, has a spatial component, namely the scale parameter, which instead of being the mean maximum distance move, it is a measure of how much the animals move away from their home range, and we extract that information from the camera trap data. And this could be attributed to differences in resident individuals or dispersing individuals, differences in how males and females use the landscape, or even differences in how seasonality affects movement. And having this scale parameter allows us to have a corrected density estimate. So with this, I ask myself, are jaguar populations increasing or decreasing? Especially when we control for change in methods through time. And if densities vary due to across their range or across environmental gradients. Now, by no means, I am not the first one to ask this question, but as I mentioned before, we have good local information. I just wanted to put this globally for the species range. And so with this, I set out 4 hypotheses. The first one, that a methodological bias between older capture-recapture and new spatial capture-recapture, namely that capture-recapture estimates would be higher than spatial capture-recapture. Secondly, that jaguar densities are declining over time, even for this method bias. Thirdly, the jaguar densities vary with the environment, and this could be attributed to different environmental covariates like elevation or rainfall. And finally, the jaguar densities vary with human influence, again attributed to different measures such as the human footprint index or forest integrity. To analyze the data, I work with meta regression models to propagate error because different density estimates have different precision or standard error. And with meta-regression, we can use the density standard error as a weighting. Therefore, we favor those estimates that are more precise, giving us a model structure similar to this, where we have the density and the method variables, the year, and environmental covariates. For the environmental covariates, I looked at different GIS layers, and I standardized them using a 25-kilometer radius buffer, and using that for each of the density estimates that we could find, and for the different environmental covariates, as well as anthropogenic, such as I previously mentioned, human footprint, landscape integrity, and also land tenure. So my data collation and standardization had this workflow where I ended up compiling 330 density estimates across about 20 years of data. And using the latitude and longitude, I was able to pinpoint those locations, create and extract those covariates, and also create landscapes, because this is important as a random effect. We could have different studies surveying near each other, but there could be nuances within those landscapes that we need to account for. And therefore, these meta mixed regression models were ideal for this analysis. And what were my results? Well, my best performing model was the one that included an interactive effect between convex polygon, the number of cameras, and differences in methodology, and including biome and human footprint. The second best model was the one that included survey year as an important covariate. But something to look at this table is that on the top 5 best performing models, methodology played a key role in determining the effect size and density. And we can see that in this chart, where studies that use spatial capture recapture have a negative effect size on jaguar densities. Convex polygon also has a negative effect size, as well as the number of cameras used. Now, the biomes was tropical grasslands and conifer forests were not significant for negative effect size, but it's still an interesting trend. But flooded savannas is where we have a higher effect size, and this is compared to tropical rainforests. Survey year had a slightly positive but non-significant effect, and human footprint did have a negative effect. So what does this tell us? So yes, methods do impact density. So study design and analysis strongly shapes densities. And this is not something that's been proved for the first time. We have evidence for jaguars, and as Matthew mentioned, for tigers as well. Are densities declining? Not really. Survey year was not included in the top model, and it showed a positive trend. And again, this could be 2 nuances at a local scale. Densities vary within the environment. They do, with the highest densities in flooded savannas, namely El Pantanal, and lowest densities in grasslands and conifer forests. And this also goes well with some studies in Venezuela that showed that the species inhabited in grassland biomes, the jaguars populations in grassland biomes, are more at risk of extinction than those ones in forests. And finally, the biggest human influence was the human footprint, and had a significant negative effect, which means the species is very sensitive to whatever any anthropogenic changes in the land. And I think this is quite interesting to, again, put it in a global scale. And this is all our tools that are learned thanks to the lab. Thank you.
Thank you, Alejandro and Matthew. Thank you, everyone. We actually are a bit over time, so have time for probably one question on Zoom, and anyone who has questions, feel free to put it in a chat and we'll send it through to the organizers, and I'll get you in touch with the speakers themselves, so then you can have discussions there. But I'll open for one question from the Zoom chat. Feel free. No. If there's no one from the Zoom, I'm just gonna read the question that's from the chat. From Adriana. Is there a direct relationship between tiger population size and diversity of prey and or habitat heterogeneity? If so, is that source of results suitable for extrapolation for top predators somewhere else? So you can read it also in the chat, Matt.
Yeah, it's a great question. We're looking at that. That answer is not available yet. We are running analyses now to look at the relationship between tigers and prey and whether or not that's positive. So like bottom up control of an ecosystem would suggest that more prey would enable more tigers. But also there's other research that shows where there's tigers, they suppress prey, right? They regulate prey population. So there might not be a positive relationship there. And how that is mediated by habitat heterogeneity, we don't know that either. So these are great questions that we are looking at in the lab and do not have answers for you yet. The other question was, are there suitable corridors for the remaining tigers in Sumatra? I would say no, but tigers will disperse through plantations and agricultural areas quite far. So there's a few parks that are so isolated that it's unlikely they will be able to be connected. And there are, for instance, Waikamba is really a great park, but it's so isolated. It's unlikely to be connected to any other forest. But the other parks that have a few kilometers, 100 kilometers of plantations between them, tigers will likely disperse through that matrix. So it's kind of called like a semi-permeable matrix between these forest patches.
Great. Well, thank you. We are going to move on to our next speaker. I would also like to announce for the speakers to make sure to look at the time. There will be a time on your screen on what you have left. We are trying to keep on time, and obviously, so we have more discussions because that's exciting. I'm going to introduce the next speaker is Dr. Maria, who will be talking about hummingbirds. And Dr. Maria has an extensive experience in research in hummingbirds with lots of publications and very great research experience there. And I will give the floor to Dr. Maria and look forward to listen to the exciting presentation. Thank you.
Tengo que estar sentada. No voy a hacer nada malo. Ya está. Pues. Can you hear me?
Yes.
Yes? Okay.
Clearly.
Well, thank you very much for inviting me, Ibrahim and Dr. Perales for this talk. It is a pleasure to be here and to share with you some of my studies in hummingbird ecology and conservation. Next one. Well, hummingbirds are birds that live only in the Americas. So in Australia, you don't have anyone. We have 59 species, 330 in the world. 13 species are endemic to Mexico, and 9 species are under conservation in the Mexican law, and 5 species threatened in the international laws. Next one. They belong to a family that is called Trochilidae, that is very homogeneous, and it's composed of neotropical birds that have long bills, very short feet, and very long wings, and very pointed wings. Next one. They are very small birds. You can see in this slide the smallest hummingbird in the world, that is called Melisuga helena, that lives in Cuba and weighs around 2 grams. And the largest one, that is called Patagona gigas, that lives in the Andes in South America and weighs 24 grams. These are very small birds. 2 grams is the weight of a grape, the grapes you eat. Next one. They feed on nectar that extract from flowers. Next one. They use for this their tongues, and the tongue is attached to the head, and it can be extracted and measured like twice the size of the bill. Next one. ¿Es un video? Pues. Eso. Yes, this is a video from National Geographic where you can see the hummingbird and how it feeds on this tube, and you can see the tongue of the hummingbird going into the nectar and sucking the nectar. You are going to see it from the above, and there you can see that it has two parts, and when it gets into the nectar, it splits to get a better time of nectar. They fly very well. You can see there the wings and the way they can stay almost stand still in front of the flowers, even when there's some wind in the environment. There you can see what they put inside the bill, and they get immediately the nectar. Next one, please. They have all the life porous bones. They never get the bones fully calcified. They have a large brain, and they have large pectoral muscles and a prominent keel, of course, to insert the prominent pectoral muscles. They have large and pointed wings. Next one. They are the best flyers among the vertebrates. They can fly in any direction. Their muscles, the flying muscles represent around 20% of the total weight. The wings can move between 80 and 20 times per second, and their flight speed varies between 50 and 95 kilometers per hour. Es un video. You can see here how they can even extract the nectar with the head up, down. Next one. The metabolism is very, very high. The body temperature is high too. It's around 40 grades centigrade. They need to feed timely, not more than 20 minutes without feeding nectar. Next one. They live between 9 and 10 years. Next one. The nests are cup-shaped. They are done with vegetable materials, and they are lined with the spider webs. They put 2 white eggs without any marks, and the females, they do all the breeding activities. Next one. This is the distribution of all the hummingbirds in the world, 330 species, 59 for Mexico, 17 for US and Canada, and all of the species in the US and Canada are also our species because they spend the winter in Mexico. They are migrants. They are pollinators. They visit more than 1,200 plant species. These are the plant species that has been reported as visited by hummingbirds. Next one. Pollination, more than 80% of the flowering plants require a pollinator, and there are more than 200,000 animals that pollinate in nature. All the systems are different, and the role of the flower visitors must be studied in detail in each system. Systems can be very generalized, but you cannot generalize that all systems are generalized or specialized. Next one. How they visit their flowers. I'm going to give you an overview of what we have been studying. Next one. We did this survey to see if there are families, plant families, where the visitation of the hummingbirds are more common, and we get to these numbers: 105 plant families and more than 1,300 species of plants visited, more than 400 species in Mexico, and almost the 300 species of hummingbirds that we have are registered visiting one or other plant species. There are many plant species, but the thing I want you to see is they love flowers with colors that are very contrasting with the environment: red, pink, orange, even white or yellow, but very contrasting with the environment. They are tube-shaped generally, but they can eat nectar from any kind of flower. They visit a lot of Heliconia plants, also plants from the genus Salvia, genus Ipomea. Next one. 31 plant families represent 80% of the registered interaction. So there are some plant families more visited than others. Next one. But if you see this with the phylogenetic of the plants and also the phylogenetic of the hummingbirds, you can see that the plants are visited almost by chance by all the hummingbirds. You can have the basal plants, the ones that are situated in the basal part of the phylogenetic tree, are more visited by more basal hummingbirds. But basal hummingbirds also visit other plants that are not in the basal part of the phylogeny. I'm going to make you an overview of some of the studies my students and I have done in Mexico. This is a map of Mexico, and the points are sites that we are going to visit as hummingbird studies. Thank you. Next one. This is the study sites varied in the diversity from 70 to 2,080 8,000 meters above sea level, from the tropical dry forest to temperate pine oak forest. Next one. This is the first one in the tropical dry forest of Chamela, where this bird that is in there, Amazilia rutila, is a dominant territorial that visits 82% of the flowering plants. There are other subordinated species listed there. Next one. Amazilia rutila use without doubt the best energetical resources, but there are some resources that are visited by other species. Next one. Because of morphological constraints. This hummingbird, you can see the very long bill, and it can visit, of course, the very long corolla flowers of some plant species that Amazilia cannot visit. So in spite of being a subordinated, this can visit any kind of flowers because of its long bill. Next one. In Manantlan, the pine oak forest is situated only 50 kilometers in a straight line from the other side. It has much more hummingbirds, 17 hummingbird species, 23 plant species. Next one. We can see here all the strategies hummingbirds use to feed. They can be dominant, they can be subordinated, but also they can be territorial, that is using its dominance to protect some patches of flowers, or trapliners, that is visiting flowers, making roots of feeding. Each species exhibit a particular behavior, but it can change with sex or with every day. Thank you. Next one. This can change seasonally. One bird can be a territorial in one season, and next season can be a trapliner. But also can change even daily because of the abundances of other hummingbirds and the abundance of this species change. Next one. The interactions have some specialization that is very good to see in this picture. You can see here a picture of a hummingbird with UV light, and you can see pollen that is placed in its head from two plant species that grow together. And you can see how the pollen is separated. The yellow one, what you see yellow in this picture, in the forehead, and the other one in the back part of the head. So plants can separate the pollen and make a better use of the visits of the pollinators. Next one. This is another site. Tehuacán-Cuicatlan Valley is covered by columnar cacti in central Mexico. Only 7 species of hummingbirds, 29 species of vegetation types. And in this site, the role of the hummingbirds are mainly nectar robbery, and because most of the plants are pollinated by bats in the night. Next one. Another site, Pine Oak Forest in Chapa de Mota, 7 hummingbird species visiting 18 plant species. Another one. El Palmito, Sinaloa. Here we did a study in altitudinal gradient from the 148 meters above sea level to above 2,000 meters. 3 vegetation types, 20 hummingbird species, 64 plant species. Next one. And in general, we can see that if there are more flowers, there will be more hummingbirds, both in species and in number of individuals. Next one. Hummingbird communities can be ruled by morphology, by plant and hummingbird abundance, by resource distribution, but the main thing is they are very variable in time and space. For conservation, of course, all of these studies of natural history are needed, but we also need to protect the habitat and to promote environmental education. Next one. These are the 5 species that are threatened under international laws. They are endemic to Mexico. Next one. And you can see here the distribution of the species. You can see the crested coquette in the upper right place. It lives only in 40 square kilometers in Guerrero, in Atoyac. And you can see the other distributions that are wider, but not so wide to ensure conservation. Next one. But hummingbirds as tigers are very charismatic. And people love hummingbirds because they represent things as love and good luck. Next one. So we can promote habitat restoration if we live in cities in the form of gardens, but if we live in the country in the form of habitat restoration. Next one. A hummingbird garden is a place where hummers can feed and rest and, if possible, can nest. We use citizen science background to promote this conservation and making tools to invite people to preserve the hummingbirds, books, leaflets, etc., and to make activities with people of all the ages, from the very beginning to the elderly. Next one. We also have worked with habitat restoration, making native plant nurseries to show people that we can produce these native plants to make the gardens. Next one. In 2014, we made the first garden for hummingbirds in Mexico City, here at the FES Iztacala. Next one. And we continue working with many schools from the high schools like this, the EMS Cuatro Tláhuac. Next one. EMS One Tlalpan, as Iztacalco. And also with kindergartens, with the conservation in La Cantera Sur. Next one. And also with elderly houses to promote the use of native plants, and in here of feeders, because they don't have the best of the vision, so it's easier. We have more than 500 gardens registered all over the country, but mainly in the area of Mexico City. You can see it in the website. Next one. Thank you. Next one.
Thank you very much for that presentation and that talk. It's very fascinating, the many different dimensions of the research that you covered, from evolution, ecology, all the way to conservation. It's amazing work. I'm going to open the floor to questions. For those on Zoom or even in the person over there at UNAM, feel free to unmute and ask a question. Alejandro, did you have your hand up? No? Okay.
No, I didn't.
Okay. All right. Any questions? Feel free to unmute yourself and ask away.
Igual si tienen preguntas en español,
Is there any from UNAM?
Yo entiendo español.
Yes, feel free to speak in Spanish. There'll be someone translating as well. So, okay, so I'm just going to go to the chat now. And everyone who's online can go and check the chat for the bios if they're interested to read more about our speakers. So the first question is, Australia is facing an issue with Varroa mite infecting bees. Is there any possibility that mites might infect native birds, like honey eaters, hummingbirds, or other species of small birds over there in Mexico?
¿Qué?
que tienen un problema con ácaros, con varroas,
Ah,
y que lo que no saben es si eso es el tipo de
varroas. No, there are no registers of varroas on hummingbirds. They are mainly on bees and honeybees and native bees, but not on hummingbirds. I think varroa, the genus varroa, is a parasite of insects, and we have never seen a varroa on hummingbirds. There are other parasites that can be transported between the countries, for sure.
Thank you. There's a question from Cindy, but I can maybe someone can read it out for me, Evelyn or Harris.
Una pregunta más de Cindy, pero está preguntando si Evelyn y Harry pueden leerla.
Oh, okay, sorry, the translation right under there. So, for those question in English is, what do hummingbirds need to nest in urban gardens? What do hummingbirds need to nest in pollinator gardens? Thank you.
Yes, to be in a pollinator garden, they need what to feed on, flowers, yes? But to nest, they need trees or shrubs where they can make the nest. They need plant material to build the nest, and they also need some spider web to tighten the nests. And that's it. If you have any tree or shrub where they can nest, you are going to find a nest. They are very easy to do.
All right, maybe we have one more question, the last one. Gracias. Amazing work. The gardens developed in Mexico City with citizen science. Are they monitoring visiting hummingbirds? How many are they visiting, and what type of gardens have been successful?
Well, we have been monitoring gardens in places like here, like Iztacala. We have a lot of students, and many of the students want to see hummingbirds. And in Mexico City, there are one species that is very common, the Berylline hummingbird, Saucerotia berylina is the more common. And the next thing to be common is Cynanthus latirostris. And we have been able to document that if you put a hummingbird garden, you will have more and more hummingbirds. If you see the gardens in Iztacala, where we have 5 hummingbird gardens, we have a lot of hummingbirds to see, and we don't use the feeders in our gardens here at Iztacala.
No,
They prefer
amazing.
the flowers, but as in the elderly houses, we use the feeders because you can put them closer to the elderly people, so it's easier for them to see.
Also makes them happy, I'm sure. Okay, well, thank you very much, Dr. Maria. Very, very interesting talk.
Thank you.
We are going to go to our next speakers, Dr. Diego from UNAM, who is a herpetologist or researching amphibians, and he's going to talk about the challenges and strategies of axolotl conservation. Very interesting. Please take the floor, Dr. Diego. Thank you. Hello. Oh, is Dr. Diego online or in person? I'm gonna wait a bit. While we wait for Dr. Diego to come and start the presentation, just a reminder. Oh, there we go.
For the presentation.
Sorry? Oh, there he is. All right. I'll let you take the floor. Thank you.
My name is Diego Chaparro. I am a research professor, and my work is sorry, I'm sorry, I'm nervous for the presentation. My work is mainly research conservation in endemic amphibians of Mexico. The genus Ambystoma is principally typical axolotl. Okay. Voy a quitar esto de ahí. Okay. Around in the world, many species is affected for the activity anthropogenic. This is for example, thrasher and deforestation and wastewater share. Okay. However, anthropogenic activities have destroyed the habitat of many species. It's okay. Sorry, I'm sorry. Es que no se alcanza a ver la presentación. ¿Te puedes hacer pequeña?
There is a translator online as well, so feel free to speak in whatever's comfortable. It is automatically real-time translated.
Yo creo que ahí está bien para que yo la pueda ver. ¿Ok? ¿Le puedes poner en la presentación para que yo empiece a avanzarla? Ok. Sorry. Okay, I'm nervous. Okay, my name is Diego Chaparro. I am a research professor in UNAM. My work is conservation in amphibians endemic of Mexico of genus Ambystoma. My topic is challenge and strategies in the conservation of axolotl endemic amphibian of Mexico. Now we speak one history of credit for lower the conservation in axolotl. The conservation in axolotl is very important for the is a species emblematic of Mexico. All the species on the planet have the right to heavy and definite environment. However, anthropogenic activities have the development in strategies for the conservation. For example, the factors for extinct in the axolotl is the trash, deforestation, and waste discharge. Placing several species in danger and extinction, as is the case of many amphibians around the world due to many particular characteristics that the present. For example, the permeable skin, the pen aquatic system for the their embryonic development, among others. In Mexico, endemic amphibians of the genus Ambystoma, axolots, are suffering this consequence. In Mexico, they laid 18 of the 13 species of axolotl, or of which 3 are threatened, 1 endangered, 11 subject to special protection, and 3 have no information. This is a lower a Mexican conservation in the stand Mexican Officer 059 is environmental protection Mexican native species of wild flora and fauna risk categories and a specification for inclusion, exclusion, or change of species hit rates. On the other hand, in the IUCN, placed 8 species in critical danger, 4 endangered, 3 in least concern, 2 white dolphins data, and 1 species not evaluation. But what needs to be done or what's behind done to protect conserve these endemic amphibians of Mexico? In the laboratory work 3 species. The first species is Ambystoma mexicanum. Ambystoma mexicanum is danger in the lower Mexican conservation in the NOM 059, sorry, and the critical danger and IUCN. Other axolotl work in the laboratory is Ambystoma altamirani, a mountain axolotl. This is a endangered for in IUCN. And finally, other axolotl work is Ambystoma lermense, axolotl from Ciénegas de Lerma, is subject to a special protection in NOM 059, and IUCN is critical danger. All axolotl in Mexico is a serious problem for the conservation. Not only 3 species is affect for the activities anthropogenic, affect the 80 species of Mexico. The ecosystem where they live have deteriorated. For example, axolotl mexicanum, axolotl mexicano, is affect signs for Hispanic times. Currently, they are cultivation areas where they use agrochemicals. The pollution, the water, currently Lake Xochimilco is a tourist area that affects the ecosystem. This activity is very bad for the conservation in the action. Some areas of Lake Xochimilco have dry hop, others are very contaminated. We found this care of wastewater share. This is a problematic for the axolotl Xochimilco. But what other axolotl, for example, the axolotl in mountain axolotl, Ambystoma altamirani, is affected for other problems. For example, the water term. In Villa del Carbón, Quilotzingo, Nicolás Romero, is the water term for the consumed human. But axolotl lermense is affect same problems axolotl mexicanum. Now the question is, can we restore what we destroyed to conserve species? Of course, I do this bioremediation. The bioremediation is very, very good, using aquatic plants for the absorption of nutrients and contaminants. In the laboratory, one project using a species native of plants. For example, Berula erecta, Polygonum mexicanum, and Polygon, sorry, Inocotyla nucleoides. This plant is very, this plant absorbs the nutrients and heavy metals. For this is important for absorbing the pollution in the lake. ¿Puedo cambiar? But the species invasive or species exotic or species alien is good. For example, the Eichhornia crassipes water jacinto as invasive plants can also function as bioremediation. In a master project, we are analyzing a biogeochemical dynamic of phosphorus and nitrogen in relationship with phytoremediation process using macrophyte. Eichhornia crassipes is a jacinto, water jacinto, and it is effect on the zooplankton community. In addition, the absorption, the plants is a refuge for zooplankton. Is important for zooplankton is the food for axolotl in the first week. This is a master research in marine science and limnology by Darío Rueda. Zooplankton is important for food in the first week developed in axolotl. Zooplankton is linked between bacteria phytoplankton and secondary consumer, and zooplankton is linked between detrital particles and secondary consumers. This is information is looked in the paper Feeding Behavior of Larvae and Vistoda mexicanum in collaboration for Dr. Nandini and Dr. Sarna. But other problem, very, very serious, is exotic species. The exotic species is introduced by human activity, threat to endemic life. They compete with endemic species and replace them. For example, the mouse and amphibian, this is introduced in America for the Spanish people. The jacinto, the Eichhornia crassipes, is introduced Brazil. But more species dangerous is carp and tilapia. Carp and tilapia, a global problem regarding introduced aquatic species are carp and tilapia in the world. They deteriorate fauna and native flora because carp and tilapia tolerate big factor physiochemical and slow factor physiochemical. But as because difference in living the axolotl and introduced species is a density in the lake. Actuality in the density in the lake is the 1 and 2 organisms for various methods. So the carp and tilapia density is a 0.032 organisms per meter square, and tilapia is 0.093 organisms meter square. So generalist organisms. All the phenomena in the lake is a typical bullying. It is a bullying ecological bullying. This aside, the fish give me your food. Axolotl native is what happens is a ecological bullying. By tolerating many environmental factors, exotic species have many survival advances. For example, the turbidity. The turbidity in lake is influencer for phytoplankton, sediments, resuspend sediments for fish, and effluent discharge. What is the effect the turbidity in the species? Increased turbidity has a negative effect on visually feeding aquatic organisms. Detect in a medium clear, detect the prey. In medium turbid, no detected the prey. Suspend particles interfere between predator and prey. So, or result, the axolotl is visually weak compared to tilapia, which has a greater visual capacity. Finding a constant disadvantage in capturing prey, since the medium water from Lake Xochimilco has turbidity that affect this capture. For example, the axolot, the area the capture operate is small compared the area capture the species invasive. The vision of axolotl is limited, only 5 centimeters away. The vision in fish is the 15 meters. In imagine, observe where area is little in clear medium for capture the air in ambystoma. Look, the big area the capture in species invasive is more. But look, the species invasive in area capture the prey in medium turbid is big or medium clear and in species native. What for what? Is for the position the eyes in the two species. The position the eyes and tilapia is the front. Is look more area. The position the eyes in species invasive is from lateral fish from ambystoma. The front is short in the look for the capture and prey. This is that the data is constant in turbidity effect. In this paper, the turbidity effect on feeding by larvae of the endemic Ambystoma mexicanum and the introduced tilapia in Lake Xochimilco Bay. But there is another very serious and dangerous problem, microplastics. The microplastic is a serious problem for what we are investigating. In laboratory, the project determination of the transfer of microplastics from predation of plankton Ambystoma mexicanum, an amphibian endemic to Mexico. As we already mentioned, zooplankton is the main food in the first weeks of development. And zooplankton feed of phytoplankton. But phytoplankton is similar to microplastics. It's a transferring the zooplankton or organisms food axolotl. This is Diana Manriquez, Master Research in Biological Science. Look the data in this paper. So other problem. This is agrochemical, mainly chlorpyrifos. And so we can continue talking about many problems that affect axolotl. For example, chlorpyrifos. But what happened warming global? It's important the temperature for axolotl. The axolotl tolerate temperature is low. When increased temperature is affect the axolotl in combination agrochemical and warming global is bad. Or the combination is the study is joint effect of global warming and agrochemical pollution on the species Ambystoma mexicanum in Lake Xochimilco. In this project is very important today. In this project, it was observed that agrochemical such as chlorpyrifos affect embryonic development. Mainly, they affect the notochorda, is the form the notochorda. Other project is affect the water quality on the feeding ecology and of axolotl Ambystoma mexicanum in collaboration on Dr. Nandini and Dr. Sarva. All problems in Xochimilco, in Villa del Carbón, in Ciénegas de Lerma, actually currently affect these 3 species of axolotl. Axolotl of Lerma, axolotl of mountain, axolotl of Xochimilco. It is very important to raise awareness among the population of this issue, environmental education. The environmental education is very important for the conservation, the axolotl in this area. Other problem is political peoples. The currently is a pretext for flags the axolotl for the all political. But with the results we have obtained, we know many doors of and political people. Because axolotl deserve to live and not be just a cultural or political flags. Concerned about the extinction of several species of axolotl, for the results obtained in the investigation, we proposed to the Congress of the State of Mexico create the first law of protection to the axolotl of Mexico. They listed them to use, and it is became created the first lower the protection the axolotl, Lake del Axolotl. It is currently in a bill that is ready to be approved. This is a special law for the preservation of the axolotl and its environment in the state of Mexico. This is a law. To achieve this, we need many collaboration and financing. For example, EY, HSBC, or two universities, one University at Tarapacá in Chile and University in Bristol in United States. Thank you for attention.
Thank you. That was an amazing presentation, and the axolotls was so cute. So many different species. I find it really fascinating that the research that is being done by UNAM is not just research, and it's an example of how you can take research into direct application and even change the law for protection. I think that is a huge learning, because often science becomes just science and then it doesn't get picked up. So this is really good collaboration and opening up. So I'll stop talking. I think we have time for 1 or 2 questions. If anyone wants to ask a question, feel free to unmute, or if you're over there at UNAM, to just step up to the microphone or speak directly, I think. Oh, sorry, there is one question here on the chat. Is there a citizen science project to work on improving water quality and habitat for axolotls in, I can't pronounce it, Xochimilco? If someone knows how to pronounce it better than I do.
Xochimilco?
Yes. Is there a citizen? Yes, is there a
Okay.
citizen science project to improve? Apologies.
No, currently, no project the conservation involucrate a society. Only institute, for example, UNAM and UAM, other institution, other countries, for example, EY, HSBC is financing the project.
Thank you. So I'm going to go directly to our next speaker. Thank you very much. Oh. Sorry, was that someone? Oh, no. Gonna go directly to our next speaker. Thank you so much for that presentation. We are introducing our next speaker from UQ, Dr. Carissa Klein, a professor. And Carissa has a lot of experience in seafood, and she has worked with many stakeholders in research, and recently also even been involved in a documentary herself. And she is going to talk to us about seafood titled, What's the Catch? Feel free to take the floor, Carissa.
Thank you, Carolina. Can you hear me and see my presentation okay? Is it all good? Yep, great. Hola, everyone from Mexico. I love Mexico. I'm really excited to be speaking to people in Mexico right now. I spent a lot of time in California and was really fortunate to be able to go visit Mexico a lot. And I would say it's definitely one of my favorite countries, definitely some of my favorite food. Here in Australia, we don't get very good Mexican food. So Thanks for having me. So yeah, today I'm going to talk about food and seafood, actually. So I'm a research scientist here at UQ, and historically a lot of my work has been done on marine conservation planning, designing marine protected areas, zoning the ocean, managing land-based impacts to marine ecosystems. That's really what I do. But in the last few years, I've gotten really excited about doing some science around the sustainability of seafood. So I'm just going to talk about what we have been up to over the last couple of years and where we're headed, just to give you a bit of an idea of what's going on. And I'd love to chat more. There's nothing specific to Mexico in here. There's some global studies and some Australian studies, but there's definitely lots of applications that we could do globally and between Australia and Mexico. So just before I start, this is here in Australia and Heron Island. This is a bunch of people from my research group and some guests and my children. So a lot of the work that I'm going to be talking about today is not just the work that I've done. I don't do a whole lot of anything other than organize people. All of these amazing people have been conducting this research and helping me do all of it. So I guess one of the biggest problems I see that we have in the world is how are we going to feed a growing population? So by 2050, it's expected that we're going to have 10 billion people on the planet, which is a couple billion more than we have now. So what are these people going to eat, and how are we going to do this with minimal impact on the environment? So huge challenge. Food in general has lots of different environmental impacts. There is a paper that estimated that food itself is responsible for 1/4 of all of the globe's greenhouse gas emissions. It takes up about half of our ice-free land to produce food and uses a whole lot of water that we use. So it has a big impact on the environment. And seafood, is part of this, and it's going to play a really important role in solving this problem of how we're going to feed the world, because seafood is incredibly nutritious. It provides a lot of the micronutrients and vitamins that we need to survive, especially in countries that don't have access to these sorts of things as easy as other countries. It can be an incredibly environmentally efficient animal-based protein if caught or grown properly, and people love it. People love to eat seafood, definitely here in Australia and globally. So seafood's going to be a really important way of figuring out how we can feed the world in an environmentally friendly way. So our research is really focusing on what are we eating, both here in Australia and globally, and where does the seafood come from? How is it produced? And what are the sustainability implications around all of those questions? So it's kind of big, and we've only started chipping away at some of these things. So I'm just going to zoom in on Australia at the moment. Seafood consumption in Australia has been on the rise over the last few decades. And the consumption of fish per capita has been increasing greater than any other food group here in Australia. So it's on the rise. Australians really rely on seafood coming from overseas rather than being produced here in our water. We're a massive island within a gigantic exclusive economic zone and big capacity to produce our own seafood, which we do some of, but we export a lot of our seafood because it's very high value. And we import, it's estimated 60 to 70% of the seafood that we consume here in Australia is imported. So that's this light blue area. And that's been, as you can see, increasing over time compared to how much we produce here in Australia. Which is fine, but there can be some sustainability implications with that, especially for other countries, which I want to talk about. So like I said, estimated 60-70% of seafood we consume is caught outside of Australian waters. So that can be in other countries' waters, and that can also be in the high seas or outside of places where countries have jurisdiction. So what are the environmental impacts of the production of seafood? So this graph is by Jessica Gephardt, who's based in America at American University. Not graph, but image. So basically, there's two main ways that seafood's produced. One's through fishing, which is on the right in the blue, and one's through aquaculture, growing the seafood, which is on the left in the green. And both of those production types are associated with a whole range of environmental impacts, whether it's to do with emissions of greenhouse gases or bycatch, like catching of species that aren't targeted. It can be lots of different pollution in terms of diseases, toxic emissions, eutrophication, land use, water use, you name it. There are a whole lot of things we need to consider when we eat seafood and if we want to sustainably produce seafood. But not only does the production of seafood have environmental impacts, it has social impacts. So when I'm talking about sustainability, I'm talking about environmental sustainability and social sustainability. So some people have found and linked global fishing and aquaculture to a whole lot of labor rights abuses and modern slavery. So people aren't getting paid properly and aren't being treated well in fisheries. And this is a really big problem around the world. And there's stuff around that. So we want to make sure that when we're eating seafood, we're not supporting that. Getting seafood from other countries increases our risk of what they call IUU fishing. So illegal, unreported, and unregulated fishing. So there's a lot of, depending on where the fishing is occurring, every country has a different sort of regulations on growing seafood, on fishing, and all of these things. Some of them have quite strict rules, others don't have any rules at all, or maybe they have rules, but they just ignore them. So a lot of fishing around the world is illegal, unreported, and unregulated, and we want to try to minimize that. Another problem with seafood is that unfortunately, we eat a lot of threatened species in seafood, and it's not illegal. So it's legal to catch, sell, and buy species that are threatened with extinction. So we found, this is one of my postdocs, this was part of her PhD, Leslie Robertson, that globally, there are 92 threatened species in global fishing records, and none of this was illegal. This was all legitimate fishing. 11 of those species are critically endangered, and this is something that's widespread across the world. 204 different countries catch or import threatened species, which is a problem. So I just have this little slide to compare what that looks like. So I have the critically endangered, according to the IUCN Red List, fish that we're eating on the left, all these funny looking species. And then I have some critically endangered species on the right from the land. So we've got the, mountain gorillas, we've got a hairy-nosed wombat up there, and rhinos, all sorts of things. So for some reason, we're quite happy to legally catch, import, sell, eat endangered species when they're in the ocean, but we're not on the land. We just wouldn't do that. It's not socially acceptable. It might happen through illegal trade, but it's not something we do. We don't go, you can't go out and order a mountain gorilla, nor would you want to, but you can go order something that's critically endangered that's in the ocean. So that's a problem. So back to what I was talking about. So this is the global map, and it shows a study that mapped out how effectively every country manages their fisheries. So basically, fisheries management effectiveness is highly variable across the globe, with the countries in red. Mexico was assessed to have ineffectively managed fisheries. This is an old study. Keep in mind that lots of places have improved since then. And then countries that are in green are very few and far between. Maybe focus on the yellow ones that have more effective fisheries management. So the reason I show this is because it shows that when a country gets seafood from another country, they're potentially getting it from a place that doesn't have great environmental and social regulations. And to just reiterate that point, so I've got Australia here in the center, and we can import seafood from countries that have poor fisheries management, so that worse off, and we can import from countries that have better fisheries management. And I guess along with that is the assumption that if we're getting seafood from countries that don't manage their fisheries well environmentally, we are sort of displacing our impacts to someone else. So I kind of calling it, you're having your cake and eating it too. So we're getting all these amazing benefits of eating seafood, but at a cost to countries, most of those countries who are disadvantaged in some way. So just to look a bit more into that, so on the x-axis here we have time, and on the y-axis we have how many tons that Australia is importing into the country. And basically it's showing that all of those top 4 red boxes so are managed to an extent that is less good than Australia. So most of the seafood that Australia gets is from a country with less effective and very ineffective fisheries management. So the dark red is really the really ineffective fisheries management. So that's problematic. Which countries are these? So this was in a little analysis that Rosa did and looked at what countries we're getting most of our seafood from. So we're getting a lot from Indonesia, China, New Zealand, etc. And it shows how much we're getting on the x-axis, and it shows how well they manage their fisheries. So that those dark red areas are that they're not managing their fisheries that well. Mexico is not on this list, but we just did sort of the top ones here. So what we found, we did a little global analysis on this, is that globally, like up to half of the seafood that all countries are importing are coming from countries that manage their fisheries less effectively than themselves. So basically they're saying, look, if you catch something here in our country, it has to meet a certain criteria and be of a certain standard, but happy to lower that standard if we get seafood from somewhere else. And this just shows on the y-axis like the management effectiveness score of every country, and on the x-axis the percent displaced from less effectively managed places, and highlighting that one big problem with this analysis is that China, who's the world's biggest exporter of seafood, has quite a high management effectiveness score, meaning that there's some debate as to whether or not that score is correct. And if that was lowered, actually these results would be much more bigger, but much more striking, because a lot of countries get seafood from China. And just to go back to this, a bit out of order, this one's, but back to Australia, everyone is always asking me, oh, what about Australia? Certainly Australia doesn't catch threatened species in the seafood, but actually, yes, we do. So Australia has at least 21, and actually there's some new data that's suggesting this is quite a lot more. 21 threatened species caught or imported by Australia. So here in Australia, we're definitely eating species that are threatened. It's not a huge amount, but it's still a problem because we have so many biodiversity regulations here, and we claim to be, a global leader in conservation, marine conservation, and we think that the whole world should do what we're doing, yet we still have some problems. So I think we need to fix these things up. And just looking at us globally, Australia is the 32 largest importer of threatened species by volume globally. And this is mainly tuna and piked dogfish. So yeah, just back to Australia, it has so many commitments to, like, the Convention on Biological Diversity, to protecting its not only our own resources, but globally making a commitment to conserve the ocean in general. And says they're a world leader in management and conservation of the environment, but yet obviously we have some work to do there. And I think looking at how we can improve the sustainability of seafood that we eat here in Australia will help have a flow-on effect in other places. here's my having your cake and eating it too. So here in Australia, we just order fish and chips like it's no big deal, not thinking about the implications socially and environmentally in other places. And I should just say in Australia, and I don't know if it's common in other countries, if you buy cooked seafood here, it only has to be labeled as fish. We don't have to know what exact fish we're eating. And obviously there's hundreds and hundreds of species of fish that we could be eating. And we would never do that for, terrestrial based meat. We never just order meat. We order chicken, we order lamb, we order beef. We don't just order fish. So that's a law in Australia that needs to change so that people can help make good decisions. Like, what fish do you want to eat? And how is that fish going? And what are the trade-offs of eating that fish for the environment and socially? So the good news is in Australia, the Australian government is, over the last couple of years, been looking at a new seafood import control policy, and we've been helping inform that policy. So they're trying to figure out ways they can get less illegal, unreported, and unregulated seafood coming into our borders. And obviously this is a global problem and issue. So it's not like we're just looking at Australia, we're looking at what are the implications of regulating imports. How does that look economically for other countries, socially, environmentally? How do we do this? So they're trying to come up with this really sort of groundbreaking, world-leading import control policy that other countries can follow suit on. Because right now we can pretty much just import whatever we want, and there's very few regulations on it in terms of seafood. And obviously there's, we can't just say stop importing or import less, because that could have also negative socioeconomic implications on the countries we're importing from, which are mostly developing nations. And a lot of this work didn't consider aquaculture. I should mention that, but we're working on aquaculture at the moment. I guess the whole goal of our research is to improve the traceability of seafood. So we know when we eat seafood, we know what it is we're eating, where it was produced, and all of that, so that we can improve the sustainability socially and economically and environmentally. And that's all from me. Happy to take any questions if there's time.
Thank you, Carissa. There is a comment in the chat about the labeling. You said it just has to be labeled fish, and there's a comment saying it's the same in Mexico.
Oh, okay. Yeah, that's good to know.
And
I'd actually love to know from Mexico as well, if somebody wants to send it. Like, we're doing a big analysis now. We're just starting it. Rosa, if she's here, is starting it on looking at legislation, biodiversity conservation legislation in every country in the world. So like, what species are listed on a country specific legislation. So like here in Australia, we have the Environmental Protection and Biodiversity Conservation Act, and we list a whole bunch of species that we're trying to protect. So I want to know what those lists are for the whole world, so we can see what we're trading.
Thank you. I'm going to start with the question in the chat, and then I'll open up to the floor. The question is, the first one here, is there a program or a way to reduce is there a program or a way to reduce and end consumption of threatened species while avoiding impacts on social stress for the community who fish them? Are those species viable for aquaculture production?
Lots of questions in there, and a really good question. So yeah, I think the problem with consuming threatened species is that I think a lot of our biodiversity conservation legislation is done separately from fisheries management and aquaculture legislation. So there's kind of a mismatch. So I think that the governments need to connect the dots there on making sure everything's a bit more consistent. So we're working on that. And in terms of are some of these species viable for aquaculture? Absolutely. I was investigating interrogating the data here in Australia. We actually definitely have viable aquaculture facilities that are producing species that are actually critically endangered here in Australia, and we're eating that seafood. So that's fine. It's grown in a farm, but there's also a slippery slope of making sure that what we're eating is from the farm and not from a wild place. So yeah, working on all of that, definitely.
Thank you. Now I'm going to open to anyone from UNAM or online. Feel free to unmute. Or, of course, in the chat if you still have questions. If not, I'll go to another one that's in the chat. What about placing posters in the fish market and fish and chip shops with the image and name of threatened species consumed in Australia? What effect does that do?
Well, I think the problem if, like, let's say if you went to an everyday fish and chip shop here in Australia and posted something and said, don't eat this species, actually the people selling the fish don't know what they're selling. They're just selling white flaky fish. They don't actually know the species. So it's really tricky, like unless we DNA tested it or you know, we wouldn't really know. So yeah, that's that probably wouldn't be an effective strategy just yet, because people don't, there's so many problems with traceability of, you know, what we're eating and what the seafood shops are selling, that I don't think we have enough information to do that just yet. But my advice is always, we have apps here in Australia, maybe in Mexico as well. We have apps that if you go to buy seafood, you can put the seafood species in the app. If you know what it is, you can figure it out. You can put in, you know, was it produced by fisheries or aquaculture? Where was it caught, farmed, whatever. And it will tell you if it's a good choice, medium choice, or bad choice. But you have to have that information to be able to put it in there. So I always encourage people to use that app. But we did another study that found that, you know, most the species that we sell here in the shops, we can't actually find the information we need to make a sustainable choice. So my suggestion and what I do is, well, if you can't figure it out, then just say no and choose something else if you have the privilege of doing that.
Thank you. Are there any questions? If there's none, before we close, actually, I have one question, Carissa.
Yes.
Is the price of fish from a well-managed country and a not well-managed country differ, or in the end, do they end up the same?
Don't know exactly, but what I do know is that places like Australia, for example, a lot of the fish that we produce here, it's very, very expensive for us to produce it because we don't have labor abuse and all of those things to the extent that other countries do. So if we were to sell the seafood that we produce here, it would be incredibly expensive. And the Australian market is unwilling to pay the price that it actually costs to produce that seafood. So we export most of the seafood that we catch because it's like really high valuable species. And the reason we import so much is because it's super cheap and affordable, and you can go down to the beach and for $10 get yourself a meal. Whereas if it was all caught here in Australia, it would not cost $10. It would cost like $60 probably. So yeah, tricky question.
Thank you.
In general, we probably don't pay the price anywhere in the country, in the world, that it actually should probably cost to consume an animal from the ocean.
All right. Well, thank you. And if there are no more questions, this is coming to the end of our session. Just some reminders before I close it. And first, after the reminders, I will send this over to the committee team because they want a group photo from all of this. But before that, for those who want to know more about the bio of the speakers who are on Zoom, they can check on the chat. For those that want to be connected to the speakers or have more questions, can feel free to contact the committee. And tomorrow, there's tomorrow and the day after, there's going to be 2 more of these sessions. The session so far has been amazing. We've been learning a lot from both sides, and I hope that this extends to a well collaboration in the future. And that's it for me, and I am going to head it to the organizing committee for the group photo, and I hope to see everyone again tomorrow. Thank you again.
All right. On behalf of all the committees of this conference, I would like to thank all the speakers today for their amazing and fascinating talks, as well as I would like to thank Carlina for moderating the session today. So before we end the session, I would like to, if you may, I would like to invite all the attendees on Zoom to turn on your camera so that we can have like a group photo. Yeah. So I'm gonna wait for like a couple of minutes. No, yeah, just maybe a minute. Yeah. Thanks for attending today. I mean, of attending this session of the 3-day conference. We're gonna see you all again tomorrow and this Friday. Yeah, I'm gonna wait for a couple more seconds for more people to turn on their cameras. All right. Okay, so I'm gonna count down to 10, then I will start taking photos. All right, in 3, 2, 1, please just hold your smile. I'll be taking several screenshots. Hold your smile. Couple more seconds. Hold your smile. All right. Okay. Yeah. Thank you all. Yeah. So we're gonna see you again tomorrow at the same time. Thank you very much.
Thank you very much, everyone.
Thanks all.